Okay, so I am back with another video about enzymes. Okay, so enzymes are proteins, and if you remember, proteins have a primary structure, they have a secondary structure, tertiary structure, and a quaternary structure. Now, remember we talked about secondary structure was things like, things like hair, keratin, structural proteins had a lot of secondary structure. And we said globular proteins like enzymes and metabolically active proteins, like enzymes, had mostly tertiary and quaternary structure. So that's where we're at. And basically, the structure of those, so like for example, here's catalase. This is an enzyme that we'll be working with next week. That's part of the experiment I want you guys to do at home, hopefully. I gotta try it out first. But this is the shape, this is the shape of catalase. And notice right here, it says active site. We'll talk about that in a moment. It says here, and its substrate is H2O or hydrogen peroxide, right? So that's why I asked you guys to check that you had that. So their shape, again, as you know, is determined by the primary structure. Remember, the primary structure is determined by the DNA, okay? And what enzymes do is they simply speed up reactions. Okay? They make chemical reactions go faster. Now, in our body, pretty much every single chemical reaction has an enzyme associated with it. We have thousands of enzymes in our body. Okay? And each one does a specific function. Okay? So generally speaking, and we'll see some exceptions to this, but they have a specific function. Like for example, catalase takes hydrogen peroxide, breaks it down into oxygen and water. All right, that's its thing. That's what it does. Okay, that's the only thing it does. If we added glucose to this, wouldn't do anything. It only works with hydrogen peroxide. So enzymes are specific, is what we're saying. All right, so that's kind of a brief introduction. Now, things like temperature, pH, can what we call denature a protein. They can change its shape, and we'll talk about that later. But it won't allow it to function. We'll get to that. But for now, enzymes, they speed up chemical reactions. They have a specific function. That is, they speed up one reaction, one type of reaction. And which reaction they speed up is determined by its shape, which is ultimately determined by the primary structure, so the order of amino acids, which is ultimately determined by DNA. So if you look here, right, if this substrate was to bump there, or there, or there, nothing would happen, right? It would just kind of bounce off each other. However, if it happened to bump right at this active site, then we got ourselves a reaction. So again, we are literally just millions and billions of random reactions happening every second. And with those enzymes, it's the shape of them that create that active site, that create that spot where the substrate can fit. And the shape is ultimately determined by the order of amino acids. Okay, so this is kind of the end of that first video. The second video, I'll talk about some of the other things in there. Thank you. Okay, and we talked about this idea before about metabolic pathways and chains, okay? So, for example, this one, and you'll learn about this later when we talk about cell respiration, but each one here, notice how that ends in ASE. This one ends in ASE. They all end in ASE. If something ends in ASE, it's an enzyme. It's a useful thing to know. So it's an enzyme. Okay? So these are reactants products. So citrate, isocitrate, alpha ketoglutarate, synchronal CoA, so forth. Those are all products or reactants. And here is the enzyme. So Alkotinase converts citrate into isocitrate. So isocitrate dehydrogenase does a couple of things here. Makes carbon dioxide, hydrogen, NADH, and this. But my point is, each enzyme has a specific function. Now, this is what we call. This is an example of a a chain. So the idea is that the products are recycled, and we'll come back to this with cell respiration. In a pathway, you get to the end, and that's all you make. Okay, so again, metabolic pathways, metabolism is literally just a series of chemical reactions. Okay, now what enzymes do is they increase the rate of reaction. So what we're talking about there is speed. They do that by lowering something called the activation energy. And basically why we have specific enzymes for each one is it allows us to control what's being produced, right? So for example, if I zoomed in on here, let's say we have enough maltase, we don't need any more. We can deactivate this enzyme and we won't make any more, right? So having enzymes at each step gives your body and your cells a lot more control over what they can do. Right, a couple things. Enzymes catalyze reactions. They're substrate specific. You might have heard this referred to as key and lock, or lock and key. And they lower the activation energy. So, for example, if you imagine, like, this is basically how much energy it takes to start a reaction. So, in chemistry, without enzymes or catalase, you'd add more. You might add heat, so you might heat up whatever you're trying to do. You might stir it to increase the kinetic energy. A lot of things you could do with that to make that reaction more likely to happen. But the whole point is, no matter what you do, you need to add that much energy. Okay? What enzymes do is they take this curve and they lower it. They make it easier for that reaction to happen. Okay? And that's the trick there. We might talk about that a little bit more later here. Okay? And basically, here's the idea of enzymes. They work by collision theory. So basically, if these are our substrates, these are our enzymes, molecules at the molecular level are moving around randomly. Okay? That's all we are, is a whole series of molecules bumping into each other, causing chemical reactions. We just do enough of those that we have life. So when this and this bump together, the enzyme speeds up that reaction and something happens. Okay? So that's the idea here. So if we make these molecules move faster, like say by increasing the temperature, then theoretically we should make more products. The enzyme should work faster. Okay? Okay, let's ignore that. Again, the collision is a result of random movements of both the substrate and the enzyme. And remember, substrate, the enzyme, and then the products. So the substrate could also, in chemistry, you would call that the reactant. Okay? So depending on how they bump into each other, that causes a chemical reaction. Right? And the successful collisions are the ones where everything gets matched up correctly. Which is kind of depressing when you think about it. But here's the idea of lowering our energy. We lower the activation energy. Okay? And really what this does is, the idea is kind of puts more stress on bonds. If we were in class, this is where I would take a pencil and I would put some pressure on it, right? So I would take my pencil I put some pressure kind of right in the middle. I cause it to bend a little bit. So I'm adding energy there, or I'm changing the shape a little bit, putting a bit more stress on those bonds, so that the remaining energy wouldn't take much just to break that pencil into two, right? Um, so the whole idea here is we're just making that reaction more likely to happen by lowering the activation energy. Okay? So this is kind of the steps involved in making an enzyme, making an enzyme substrate reaction. So the substrate has to combine with the active site, right? So this is where the activation energy is lowered, okay, at the active site. And this has a specific shape. And this shape is going to match the substrate. So you see down here, A, B, and C, they all have a very specific shape. B can only fit in this one, C can only fit in this one, A can only fit in this one. Okay? So that's kind of the first step. Remember, it has to be in the right orientation. If A hits here, nothing's going to happen. So then what you end up there is you get what we call the enzyme substrate complex. And that's what this picture calls the transition state. So you get that complex. At that point, those stress on those bonds causes that to shift around, to shift and to break. So the chemical reaction takes place. So in this case, this bond's broken and this bond's formed, and everybody goes off, changed, right? So C now we have some product called A and B. But notice here, the enzyme, there's no change, right? So the enzyme could go on, find another A and find another BC over here. It can do the whole reaction again. Okay, so it's pretty convenient that, in the sense that the enzymes can keep catalyzing more of the same substrate. Okay, so one model of enzymes, one way we think enzymes work is what we call the lock and key model. Okay, so here the substrate and the active site match perfectly. So the idea is one enzyme, one substrate. So here, A can only bind with A, B can only bind here, C can only bind here. It's a lock and key model. And again, you kind of figure out why it's called a lock and key. You know, if you have a certain key, it only opens one lock. So it's very specific. Okay, so that's the first model. The enzyme, these as the active site. And that's where the chemical reaction takes place, okay? So that's where the chemical reaction takes place, okay? So that's where it's sped up. There's a second model that we call the induced fit model. So what you can see here is you see site A, B, and C, and the substrate doesn't perfectly fit there. But as that substrate gets closer, the forces and the bonding, the hydrogen bonding, the van der Waals forces, all those wonderful forces we've been talking about, cause that enzyme to change its shape slightly so that it all matches, it all ends up working perfectly. So the idea here is the substrate binding causes the active site of the enzyme to change shape a little bit. Okay, I always think of this as putting on a glove. Okay, you can put on the glove, but you gotta, you've kind of got to pull it and shift it and get it to fit just right. It's not a perfect fit as soon as you put the glove on. So what this means is you could have a substrate like this, or if you can imagine, I make a great drawing here that's exactly like this, but the top's like that. That would probably be just fine. It would work okay in an induced fit model. May not work so well in a lock and key model, but different enzymes are adapted a little bit differently for that. Okay, so I hope that kind of explains the basics of how enzymes work.